Okay, this is chapter 27 on bacteria and archaea. It's about 18 or so pages in your textbook, and I'm going to try to run through this a little bit quickly to keep the file size low, but you're obviously able to stop and pause or do whatever you want, and this will be on the next assessment. So, obviously, bacteria and archaea are both prokaryotes, and these are basically one of life's two main groups. We have the prokaryotes and we have the eukaryotes. And as we said before, the prokaryotes are actually paraphyletic, like we said when we talked about phylogeny, so meaning prokaryotes are not a monophyletic group. Well, we still talk about them as if they were, although erroneously. And so prokaryotes within them are the bacteria and the archaea. Like we said, bacteria are basal, and so they are the outgroup when it comes to all of life. And so as we said down there, that should actually say paraphyletic, not polyphyletic, but either way, it is not a monophyletic group. When it comes to prokaryotes, they have huge abundance. They are the Earth's most abundant organisms. Prokaryotes are everywhere, on every continent, in every habitat you can think of, even on you. But largely, the taxonomy of this group is basically unknown. There are not many researchers researching bacteria taxonomy, and so it remains to be seen what's really out there when it comes to diversity. There are approximately, this is a big approximation, 5 nonillion bacteria alive today. When you look at nonillion, there's nonillion, a lot of zeros. And there are probably millions and millions of species, but only about 5,000 have been described by taxonomists. As you can imagine, there's not much difference morphologically between one bacteria and another, or at least the majority of them, and so describing these morphologically is difficult. You kind of need modern genomics and modern phylogenetic techniques to do so. So, as I just said, they're usually based on the MSC, described based on MSC because they're looking at how they look, but more recent work has gone into more PSC, TSC, PSC being phylogenetic species concepts. So, even though there are lots of different bacteria, there's not much known about them, and that goes with the nutritional modes as well. There's just not much known about what they do. But we have a few categories of prokaryotes, and so, for example, we have photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. The first word, photo and chemo, is basically talking about where the energy comes from. Is it, do they get it from the sun, or do they get it from chemicals in their environment? The second word there, the infix here, auto or hetero means do they get their carbon from the outside environment, which would be hetero, or do they get it, can they make it themselves? And so because we have these two different prefixes, there's 4 different possibilities: photoauto, photohetero, chemoauto, and chemohetero. And there are prokaryotes that do all 4 of these things, or any of the 4 of these things. Plants, for example, most plants are photoautotrophic. They get their carbon from carbon dioxide, they get their energy from light. And then this also includes us, animals, and protists are chemoheterotrophic. Let's see if I can expand this a bit. Nope. Okay. Never mind. So when we look at the structure and function of our prokaryotes, most prokaryotic cells are unicellular. In fact, not most, all. All prokaryotic cells are unicellular. All of them are very small, much smaller than eukaryotic cells. They do not have a nucleus. They do not have any organelles at all. And instead of a nucleus, they have a region in their cytoplasm called the nucleoid. The nucleoid is just a region of the cytoplasm where the DNA sits, and it basically serves the function of the nucleus in eukaryotes. They have one chromosome, and that one chromosome is circular. okay, unlike eukaryotes where we have a linear chromosome. And they have structures, DNA structures called plasmids, and plasmids can be transferred between bacteria. And remember when we talked about horizontal gene transfer, and we said that genes were passed between prokaryotes within the same generation? Well, this is what they're passing. They're passing those plasmids between each other, and those plasmids hold genes that can then be incorporated into their single chromosome. Okay? Okay. Also, all prokaryotes have a cell wall, okay, unlike animals, obviously, where we do not have a cell wall. And also, this cell wall is different than a plant cell wall. It is not a cellulose-based cell wall that plants have. It is a peptidoglycan-based cell wall. Peptidoglycan is just a large sugar. And they have ribosomes, basically protein-making factories. And they have a flagellum or a pilus. Flagellum is going to be a structure that is going to be allowing for movement. Pilus also allows for movement as well, but can also be used in that plasmid transfer that we talked about a little bit ago. So the body plan of prokaryote is pretty simple. Notice we have in the middle here, we have the nucleoid, and then we have just a part of the cytoplasm, and then we have just exterior to the cytoplasm, the cell membrane. And then just exterior to the cell membrane, we have a cell wall. And then oftentimes exterior to the cell wall, you have something called a capsule. And we'll define this a little bit later, but it's basically a structure that's going to be helping bacteria interact with each other and form something called the biofilm. Okay? And then we have, as you see in the diagram, we have flagella, and sometimes we have pili, singular being a pilus. That sometimes helps in movement, sometimes helps with transferring plasmids between bacteria. So we have some general prokaryotic cell shapes. On the left, you have a coccus-shaped cell, basically a spherical-shaped organism. And notice in that picture we have somewhere like 20 or 30 different organisms all grouped together. So every single one of them is a circle-shaped. In the middle here, we have bacillus. Bacillus is a rod shape. So in that diagram there in the middle, we probably have, 50 or so bacillus bacteria all grouped together. And then on the right, we have spirillum, which is a spiral-shaped bacterium. And we just have one spirillum bacterium in that picture there. Prokaryotes reproduce in several methods, so either by vertical gene transfer or horizontal gene transfer, like we talked about in the phylogeny lecture. So vertical gene transfer meaning they pass down their genes to their ancestors via reproduction, either sexual or asexual. And then there's horizontal gene transfer, which, like we talked about earlier, is basically passing genes among bacteria in the same generation. Okay, so for vertical gene transfer, there's only one method, and that method is binary fission. Remember from the first week, bacteria in archaea, the prokaryotes, do not use mitosis. They do not use meiosis. This is what they use to reproduce, binary fission. For horizontal gene transfer, there's 3 different ways that prokaryotes can reproduce. Number 1 is going to be conjugation. Conjugation is where they're transferring that plasmid that we talked about between cells, and they do this via a pilus, and it's a special kind of pilus called a sex pilus. Basically, the sex pilus is like a hollow tube. It connects 2 bacterial cells together, or archaeal tubes, any prokaryote, bacteria or archaea. It connects those 2 cells together, and then the plasmid is sent through that tube, and then it can incorporate those genes from that plasmid into its chromosome. So that's called conjugation. Number 2 is transduction. Transduction is where they can't really have any control over this. It just happens. A virus, as you can see in that diagram, called a bacteriophage. A bacteriophage is a special type of virus that only infects bacteria. The bacteriophage lands on the prokaryote, injects its DNA into the prokaryote, and sometimes this DNA actually gets incorporated into the genome, and the new genes are incorporated into its body. And then the last one here is transformation, where basically bacteria or archaea wander along, find a gene randomly in the environment, and then pick it up and incorporate it into their genome. Oftentimes, this is when bacteria are grouped together, and genes are sort of plentiful in the environment, and then they're picked up and incorporated, usually in the form of plasmids that are in the environment. Okay, so those are the three types of horizontal gene transfer that prokaryotes use to reproduce. So the bacteria themselves, let's go through a little bit of taxonomy when it comes to the bacteria. Bacteria are the oldest organisms on Earth. They are the most diverse organisms on Earth, and they are the most abundant organisms on Earth, period. Okay? They are also the most oldest, diverse, and abundant of the two prokaryotic lineages. Bacteria are mostly harmless, at least when concerning us. They basically are not going to be hurting us, most of them in any way. Some of them are even beneficial, right, by releasing oxygen into the environment, doing something called nitrogen fixation, where they take nitrogen from the atmosphere, incorporate it into other molecules, such as nitrate, nitrite, or ammonia, and then those chemicals can then be taken up by plants, because plants cannot take nitrogen from the atmosphere. So they need bacteria to do that for them. So all these are very beneficial things. Only a few bacteria are actually harmful. Okay, only a few bacteria cause disease, and those disease-causing bacteria are called pathogens. Okay, bacteria also talk to each other and can communicate by a process called quorum sensing. Okay, quorum sensing is where bacteria are grouped together, and but of course they have no eyes, they have no ears, they have no way to sense the environment really, except for chemically they can sense the environment. So every bacterium releases some chemicals, and then the other bacteria in the environment release chemicals as well, and then they all detect how many chemicals are there, which indicates how many bacteria are there, and then once that chemical level reaches a certain threshold, they all act together and do some type of event. So let me see if this video that talks about quorum sensing here will actually play. Guess not. Okay. I'll put that video on ACE so you can watch it later. Okay, so let's talk about the 5 main bacterial groups. And the first one here I'd like to talk about is called proteobacteria. Proteobacteria are going to be the most diverse group of bacteria that we know of. Lots of different feeding modes. There are photoautotrophs here, there are chemoautotrophs here, and there are chemoheterotrophs within the proteobacteria. So a lot of them can make their own food, and a lot of them can make their own energy, and some of them get those things from the environment too. Within proteobacteria, there are some important ones we need to talk about, including the proteobacteria that do nitrogen fixation. Remember I mentioned that nitrogen fixation is basically where an organism, usually bacteria, takes nitrogen from the atmosphere and incorporates it through its body into chemicals such as nitrate, nitrite, or ammonia, and then it releases this into the soil, or in some way plants take this up. Sometimes the bacteria are in little nodules within the roots of the plant, like you can see in this picture. Those little spheres that are on the roots of this plant, it's a legume, are actually filled with nitrogen fixing bacteria. Basically for the plant, this is a nitrogen making factory for them. It puts it into one of these forms that the plant can take up. So very important. But proteobacteria also include other important bacteria such as E. coli, which is usually very helpful for digestion for us and other mammals as well. Some can be harmful, but most E. coli is going to be helpful. Salmonella, most of which not so helpful, at least what you hear about. The ancestor to all mitochondria in all vertebrates is a proteobacterium, or was a proteobacterium. Okay, and also the cause of the disease cholera. So Vibrio cholerae is a proteobacterium. And finally, you've heard of the Black Death or the Black Plague that affected the Middle Ages hundreds and hundreds of years ago. That was caused by Yersinia, a pathogen that is a proteobacterium, and that causes the plague. The plague is still around. It's not something that was then and not anymore. We just know the cause of it, and it sometimes pops up in certain communities. So Yersinia is still around. We just have the medical wherewithal to control it nowadays. Okay, so all these are proteobacteria.
For the first time, scientists discovered DNA of the bacteria that caused London's devastating Black Death in the 17th century. A recent breakthrough in plague research has identified the bacterium in 5 different skeletons recovered in 2015 from a suspected Great Plague burial pit at a modern construction site. Experts took teeth from 20 individual skeletons and sent them to Germany's Max Planck Institute for analysis. The group found DNA from the Yersinia pestis plague bacterium in 5 of the 20 samples. A statement from the Museum of London reads, quote, "The enamel shells of the teeth protected and preserved the DNA, essentially acting as little time capsules, providing scientific evidence that these people had been exposed to the bacterium." The bubonic plague broke out in London in 1665 and killed an estimated 100,000 people. Scientists now hope to further analyze the bacteria's DNA to understand how the plague evolves and prepare for potential future outbreaks. For Newsbeat Social, I'm Cameron Hallwell.
So as you can see there, Yersinia does cause the plague, and it was found in plague victims, and it still pops up now, here and then. Our second group are the chlamydias. Chlamydias are basically going to be chemoheterotrophs. So they are always going to be parasites of vertebrates, including humans. And they share a common ancestor with a cyanobacterium. Okay, we talked about cyanobacteria when we talked about the snowball Earth, something that happened billions and billions of years ago, is it million? I think it's maybe millions of years ago. But the cyanobacterium is basically going to be a oxygen, a photosynthetic bacteria that's going to be releasing oxygen in the atmosphere. And so chlamydias are an ancestor of cyanobacteria. So there you go, right there. Cyanobacteria are very plant-like. And so because of that, chlamydias, in a lot of ways, have some plant-like characteristics. So they diverged from the cyanobacteria about 1 billion years ago. They're very, very small. As you can see this image here, this is within cells. And so these guys are inside of cells, so they have to be very tiny, so they're infecting cells within vertebrates and other organisms. Chlamydias include, of course, the chlamydia sexual transmitted disease that we know about. Chlamydia is going to be the cause of that, and also some forms of pneumonia. Chlamydia also causes those. Our third bacterial group are the spirochetes, and spirochetes, just like its name suggests, have a spiral shape to them. They are also, just like our chlamydias, chemoheterotrophs, so a lot of them are going to be parasites of other organisms. Some of them are free-living, some of them are anaerobic, so in other words, they have to live in oxygen-poor environments, and we have some parasites as well. They cause some diseases, so they can be pathogens, and they cause diseases that we are familiar with, such as Lyme disease, which is carried by ticks, right? And so inside of the ticks, in their digestive system, is the spirochete, and this spirochete, when injected into a human because of a tick bite, can then cause Lyme disease. Spirochetes also cause syphilis, and there is some research to suggest that some forms of Alzheimer's disease may be caused or their conditions exacerbated by a spirochete. So there's ongoing research that is studying the link between spirochetes and Alzheimer's disease. Our fourth group here are the gram-positive bacteria. They are called this because they they stain a certain color when they're stained by a stain called gram staining. And this is because of their unique morphology where they have very thick walls. Most of these are chemoautotrophs, and so many are going to be important decomposers within the soil, and they're going to be breaking down organisms, and a lot of them are also going to be breaking down food within the guts of vertebrates, including you and I. For example, yogurt. The cultures within yogurt include gram-positive bacteria, and then there are some gram-positive bacteria that cause deadly diseases, so that in other words act as pathogens. These include listeriosis, it also includes anthrax, and it also includes food poisoning, also tetanus and botulism. And so tetanus is obviously going to be caused by the bacteria that usually are present on oxidized environments such as rust, and botulism is usually caused by bacteria that are within spoiled foods, and oftentimes this is evidenced by bulging aluminum cans, and botulism can be very, very dangerous. Our last group here are the cyanobacteria. Cyanobacteria are commonly called green algae. They are photoautotrophic, so they do go through photosynthesis. They make their own energy. They get carbon from the atmosphere. As we said before, these are the ones that cause that first mass extinction of all the anaerobic bacteria on Earth, because they put out so much oxygen that these anaerobic bacteria could not survive in an aerobic environment. Okay, remember, oxygen is the waste product of photosynthesis. So they went through photosynthesis, produced oxygen, and therefore made the world full of oxygen. So thank you, cyanobacteria. We would not be here if not for them. Cyanobacteria also were the ancestor of chloroplasts, right? We said through endosymbiosis, not we, of course, but plants had chloroplasts inside of their bodies, and this chloroplast was from a cyanobacterium. They also go through nitrogen fixation, and they are part of what we're calling phytoplankton. Phytoplankton are basically microscopic organisms that live on top or just shallow areas, or not shallow areas, but the surface of ocean environments, lake environments, and commonly are filled with a lot of protists too. So we'll talk about phytoplankton again when we talk about protists, but cyanobacteria are a big part of that. Cyanobacteria can also be called algal blooms. Basically, when you have an excess amount of nutrients in a water system, the algae basically take advantage of those high amounts of nutrients and cause something called a bloom, where they reproduce at massive amounts and therefore cover the entire lake or river or stream or whatever it is in algae. When that happens, a lot of the algae eventually sink to the bottom and die, releasing carbon dioxide and also taking and sucking oxygen from the environment, often causing fish deaths, something called a fish kill. And so fish kills can usually be associated with algal blooms. So that's enough for bacteria. Let's move on to talk about the taxonomy of archaea, our second prokaryotic group. And so archaea were thought to be bacteria until around the late 1970s, where some research was shown that they are in fact a separate group. They split from bacteria around 3.8 billion years ago. So they have not been related to bacteria for a long time. In fact, archaea are more related to us, eukaryotes, than they are related to bacteria. If you go back and look at that phylogeny on that first slide, you'll see that eukaryotes, us, and archaea are sister taxa. The bacteria are the outgroup when it comes to life. Okay, archaea are everywhere. They live in common environments, forest habitats, prairie habitats, marsh habitats, desert habitats. They live everywhere. but they're commonly associated with extreme environments. So they're commonly called extremophiles because a lot of them live in very hot or very cold or very salty habitats. And so, therefore, they're erroneously associated with extreme environments, though in reality, they can exist in many, many, many different habitats. One interesting aspect of archaea is that there are no known archaean pathogens. No archaea infect humans and give or cause diseases in humans. Okay, so only bacteria do that. When it comes to the prokaryotes, the archaea do not. So a few archaean groups, and these are generally separated by their feeding modes. Okay. The first one here are the methanogens. And methanogens, as the name suggests, produce methane. And they do this as a waste byproduct. And so methane CH4, this is basically put into the atmosphere. Oftentimes these organisms are underwater, so they're releasing methane. And as you see in that image there, the methane's coming up as bubbles. Oftentimes this can be seen in marshy or swampy habitats. This is why marshes and swamps sometimes smell really bad, is because the methanogens are living within the water, pushing that methane up, and it breaks the water surface and then makes it smell pretty nasty. Methane is not a good smelling chemical. So methanogens are strict anaerobes. They have to live in oxygen-free environments. And they live in lots of places. As I said, marshes and swamps is one of them, but they also live in the guts of many organisms, including a lot of terrestrial organisms such as cows and livestock have methanogens within their guts, which is why cows, basically when cows are processing food, the methanogens are helping them break down that food, but they're also going to be pushing out a lot of methane, and therefore that's also going to have a negative effect on the climate, because methane is a very, very strong greenhouse gas. Just even more so than carbon dioxide, methane traps heat within the atmosphere. Okay, so the more cows we have, the more farting happens. That's literally what it is. And therefore, the more methane is produced in the atmosphere. And it has a significant effect. As I said, marshes, Antarctic ice has some methane producers, the ocean, and also geologically there are some rocks that hold methanogens as well. And as we said, methane gas is basically a byproduct of their metabolism. They don't use it, they're just producing this as a waste. Just like the cyanobacteria was producing oxygen as a waste, methanogens produce methane as a waste. Second group of archaeans are the halophiles. The halophiles are basically salt-loving organisms, and these guys are either they're either aerobic or oftentimes photosynthetic. They love to live in things that humans have created, called evaporation ponds. And saltwater evaporation ponds are used by companies that are basically growing salt crystals. And they'll put out seawater, have the water evaporate, and what's remaining is the salt from the seawater. And of course, halophiles, loving salt, love these saltwater evaporation ponds. As you can see in the background there, they'll start to inhabit these ponds. And because of their morphology, either being orange or red or green, whatever color they are, it turns the entire saltwater evaporation pond that color of their bodies. Great Salt Lake has a lot of halophiles. The Dead Sea, also very, very salty, lots of halophiles as well. Our third group are the thermophiles. The thermophiles love hot environments, and these are usually either chemoautotrophs or heterotrophs. Oftentimes they live near hydrothermal vents, and so, for example, at Yellowstone National Park out west, they have these geysers and hydrothermal vents that are very hot, as you can see in the background of this image, and then all of the surrounding underwater area there is just completely inhabited by these archaeans, these thermophile archaeans. They love very hot temperatures, 230 degrees Fahrenheit. Excuse me. So even that's above boiling, and they love these environments. They also love living in hot springs, environments that have very hot water coming out of them. You can find these. Hydrothermal vents underneath the ocean are also areas where these thermophiles love. The opposite of that would be the psychrophiles. The psychrophiles are cold-loving archaeans. Most archaeans that are extremophiles are psychrophiles. So this is sort of the most common extremophile archaean type. Psychrophiles basically have a type of antifreeze-like substance within their cells that prevents them from freezing and cracking and breaking within extreme cold weather. This protein antifreeze has also been found to be in some arthropods as well, so obviously animals. And so it may be a similar protein that helps a lot of cold-loving and cold-active arthropods survive in Canada and Siberia and other northern parts. They can survive extremely cold environments, negative 45 degrees Celsius. Obviously, that's way below freezing. And apparently they just love these environments. So they inhabit a lot of the colder regions of Earth, such as the alpine and polar habitats, like I was mentioning, northern Russia, northern Canada and Scandinavia, Greenland. They love the deep ocean where it's extremely cold. And they also love cave environments where it can get quite cold as well. They also have been found in the upper atmosphere. And so this is another very cold environment where they'd love to survive in. So a little bit more, we talked about the taxonomy of these groups. Let's talk a little bit about the ecology of these groups. So the role of prokaryotes in the biosphere is an extremely important one. In fact, if you ask the question, what would happen to the Earth and to the abundance and success of other organisms on Earth if humans disappeared? Well, they'd probably be better off. Most human-caused environmental conditions are of distress, damage, and altering so that other organisms have an environment that they cannot live in. So if we were gone, probably most animals would be more successful and would be thriving at higher levels. However, the same cannot be said about bacteria. If bacteria in archaea, if prokaryotes were gone, we'd all be screwed. All life would die, almost likely within a few years. This is because prokaryotes play such a critical role in the environment that we could not live without them, and most animals and most plants and other forms of life also could not live without them. Let's talk about what it is prokaryotes do that makes them so critical for not just their survival on Earth, but all life survival on Earth. Number one is called chemical cycling. Chemical cycling is basically just the recycling of chemicals between the biotic realm and the abiotic realm. So biotic meaning living, abiotic meaning non-living. So this basically means breaking down dead organisms and turning it into more basic chemicals. Breaking down corpses, breaking down waste, vegetation, leaf litter. Think of every autumn, all the leaves that fall from trees. All that has to be broken down. Yes, protists do some of that. Earthworms do some of that. Fungi does some of that. But none of those organisms do as much as bacteria do. Okay, bacteria are the number one, the main decomposers of almost every ecosystem on Earth. They also go through nitrogen cycling, and we talked about this, nitrogen fixation, right? Nitrogen, same as nitrogen cycling, basically incorporating nitrogen from the atmosphere into their bodies, into chemicals that can be taken up by plants, and therefore plant life can survive. If you had no bacteria, you would have no nitrogen available for plants. All plants would be dead. All animals would be dead. Bacteria have also been shown to increase the amount of available nutrients in soils. And so the most critical nutrients for plant survival are N, P, and K, nitrogen, phosphorus, potassium. And there's evidence of this if you look at, for example, any fertilizer bag, it'll tell you the NPK rating because that's the most important things for plant growth. When prokaryotes are in the environment, they increase the levels of N, P, and K in the environment, so they increase nutrient availability for other organisms. Also, prokaryotes have a lot of interactions, direct interactions with other organisms. For example, they go through a lot of symbioses. Symbiosis is basically a relationship where two animals living together. It doesn't necessarily have to be good. For example, parasitism is a type of symbiosis, right? But oftentimes it is helpful for both organisms, in which case it's called a mutualism. So symbiosis is sort of a general overall term for all of these interactions, whether good or bad. It just means organisms living together. When prokaryotes are involved, prokaryotes are going to be doing one of many of these things. So for example, some prokaryotes are mutualistic with other organisms. As you see in this picture, the prokaryotes are living on this fish, and these are going to be prokaryotes that go through a biofluorescence activity, and so helping the fish see in very deep waters, but also the prokaryote is going to get nutrients from the fish as well. So a mutualism where they both help each other out. We have commensalism. Commensalism is where we have one organism being helped by the relationship and the other organism not affected by the relationship. And then there are some, as we mentioned earlier, some prokaryotes that cause disease. Remember, only bacteria cause disease, so pathogens would be parasites, okay? A disease-causing parasite. Only bacteria, not archaea. And lastly, let's talk about our relationship to prokaryotes. Some prokaryotes are human pathogens, right? In fact, prokaryotes cause half of all human diseases in the world. However, we could not live without prokaryotes. Prokaryotes live within our guts. They live on our bodies. They help us survive. They break down the food inside of our bodies, and so we could not survive without them. This is why humans and other vertebrates are often called superorganisms, and you can look at the superorganism scale. What a superorganism is when you take into account all of the bacteria that allows us to live. It's not just one organism when you look in the mirror. It is all of the bacteria that you can't see and yourself. That is the superorganism view. If you broke it down by, for example, number of species that make up this superorganism that is you, as you see in that diagram, the blue represents bacterial species. That little tiny sliver of pink, or in this case white because you can't even see the pink, that represents Homo sapiens, you. If you looked at the number of cells, again, the blue represent bacterial cells that make up you, superorganism, and the pink represents the number of human cells that makes up you at the superorganism scale. So, for example, if you looked at the human body and took a test swab of what prokaryotes exist on the human body, you would get a different diversity of prokaryotes for swabbing different areas. And every single one of these prokaryotes has evolved to the very specific micro-environmental conditions of that part of your body. The pH, the temperature, what chemicals are there, how much oil is there, how many hair follicles are there. Every single particular or environmental condition on your body is adapted to by those prokaryotes. And lastly, of course, we have the toxic pathogens. And so we have two main types of toxins. We have exotoxins and we have endotoxins. So exotoxins are those toxins secreted by bacteria actively. Okay, so for example, cholera. Vibrio cholerae is the bacterium that causes the disease called cholera. Vibrio cholerae actively excretes a toxin into its host body to cause cholera. The opposite of that would be an endotoxin. An endotoxin is not actively secreted by the bacterium. It's just within the bacterium's body, and then when the bacteria dies, the body breaks apart and the toxin leaches out. So it's not actively secreted. An example of this would be typhoid fever, caused by Salmonella typhi. Salmonella typhi releases an endotoxin upon its own death, and that toxin causes typhoid fever. So it's sort of like an accidental causing the pathogen or causing the disease. Lastly, prokaryotes are also critical for humans in research and development. Prokaryotes are the principal agents in something called bioremediation, where we use prokaryotes, for example, to clean up the environment after an environmental disaster. For example, as shown in the picture here, an oil spill. What researchers commonly do is they spray oil-consuming or oil-eating bacteria onto these rocks and onto the area that's affected by the oil spill, and then over time these bacteria break down all the hydrocarbons and other oils and nasty chemicals in that environment. And so that's called bioremediation, basically cleaning up the environment using life, bio, in this case the bacteria. Prokaryotes are also used to create certain things. They're used to create antibiotics, they're used to create vitamins, they're used to create hormones, they're even used to create plastics. And this is by incorporating genes into the bacterium. Usually this is bacteria. We incorporate genes into the bacteria that do a certain thing. For example, you have a gene that creates vitamin B. You want more vitamin B. You incorporate this gene into the bacteria, let the bacteria reproduce rapidly at a huge, huge rate. Suddenly you have millions of bacteria that can produce vitamin B. Give them food, let them do their thing, harvest the vitamin B. And so we use bacteria to do this on a massive, massive scale. Okay, I think that is it. So I will see you tomorrow, or I guess if you're watching this early, I won't, but I'll see you all back on Wednesday, and we'll continue with a lab, and that's it for now. See you.